Chapter 14 is all about gene expression, how a gene is made into a protein. The first concept in the chapter is how the gene itself, and what is a gene, how it specifies proteins via the process of transcription and the process of translation. So what exactly is gene expression? Gene expression is the process by which DNA directs the synthesis of proteins or RNA. The old idea of that process is that one gene directed the synthesis of one enzyme. This was proposed by Beadle and Tatum, and what they did experiments with was mold. Now, the reason that they chose to use mold as opposed to fruit flies, for example, mold is haploid. So there's only one set of chromosomes. So they could easily change the allele, one allele, and see the outcome or change or mutant in the offspring directly. Whereas fruit flies are diploid, we would have to look at homozygous dominant, heterozygous, homozygous recessive. In the case of mold, you make one change and you're going to see it in the phenotype. There's no hidden alleles. One allele, one change in the phenotype. Now, what they did was they grew mold on something called agar. So what agar is it's a type of solution, and it turns into a gel, and it's made of salt, sugar, different kinds of vitamins. And they exposed different molds to X-ray radiation. Now, by doing this, they could then grow the molds in different mediums or different agar plates, and they could see how it would grow in a different environment. So from that, they figured out that the function of the gene dictated the production of a specific enzyme, depending on if it was able to synthesize or metabolize the enzyme in the agar plate. So the problem with their experiment was that not all proteins are enzymes. So a newer idea is that one gene and one polypeptide synthesized. The most accurate description of gene expression is that one gene to one RNA molecule, which is down the road going to be turned into a polypeptide. Now, something that I should mention is when you see polypeptide, what a polypeptide is it's a chain of amino acids. Remember, amino acids put together make a polypeptide, and when you get two or more polypeptide chains all thrown about, that is what a protein is. So the flow of genetic information and the theme of this entire chapter is that DNA indirectly makes RNA, which indirectly makes protein. What this is known as is the central dogma of biology. DNA codes for RNA, which codes for protein. This was figured out by Francis Crick, and the actual central dogma is going to take place in two steps. A process known as transcription, which is DNA is read and RNA is synthesized. Translation is where the RNA is read and a protein is put together. Now, where is the protein synthesized? Where is it put together? It is put together at the ribosome. This is the site of translation or the site of protein synthesis. So how does the central dogma work in a eukaryotic cell versus a prokaryotic cell or a bacteria cell? The process of a DNA being read and RNA being made occurs in the cytoplasm of a prokaryotic cell or a bacteria cell. At the same time that that is taking place, a ribosome is actively building the polypeptide or the protein, which again is a process known as translation. So again, DNA is going to essentially make the RNA, which is going to then code for that polypeptide. All this can be occurring at the same time because there's no divider, there's no nucleus inside that bacteria cell. So as soon as that strand of RNA is made, it can be read and a protein or polypeptide can be made. Now in a eukaryotic cell, like a cell like ours, it's a little bit different because there's a nucleus. And that's where the genetic information is, and that's where the DNA is trapped. So the process of transcription, where again the DNA is read and RNA is made, occurs within the nucleus. The RNA, being a single strand, is able to leave the nucleus through a nuclear pore, go into the cytoplasm, and make its way to that ribosome in order to be read and the protein to be synthesized. Again, a process known as translation. Please remember, one gene equals one RNA molecule. So the difference between DNA and RNA, a little bit of review. So DNA is made of nucleotides. Nucleotide is a phosphate molecule, a sugar, deoxyribose, and a nitrogenous base. A DNA molecule is a double strand. Again, the sugar is deoxyribose, and one of the nitrogenous bases is thymine. DNA is the template for this process. RNA is a nucleic acid as well, and it's also made of nucleotides, but it is a single strand. The sugar in that nucleotide is different. It is ribose. Instead of thymine, there is no nitrogenous base thymine in RNA. Instead, it is uracil. And RNA has many, many different roles or jobs within our cell, whereas DNA, again, is just the template to be read or the directions. All the different types of RNA that are in our cell. So in a eukaryotic cell, there is something called pre-mRNA. This is the precursor to mRNA. This is brand new RNA that has been transcribed via transcription, but it hasn't been edited for mistakes yet. Once it has been edited for mistakes, it is called mRNA. So this is the edited version, and this is going to carry the code from DNA and specify what amino acid is going to go into that polypeptide. Another type is tRNA, and this type of RNA has a job of carrying a specific amino acid to the ribosome in order to build that polypeptide. And we'll get into what is an anticodon and codon later. Another type is rRNA, which is ribosomal RNA. So this makes up 60% of the ribosome, and this is where the protein synthesis or building of the polypeptide takes place. A new type is called snRNA, and this is small nuclear RNA, part of a spliceosome, structural and has catalytic roles within our nucleus. Another type is srpRNA, and that stands for signal recognition particle, and it binds to signal peptides. Another type is RNAi, and this is a interference RNA, so it regulates. Another type is miRNA or siRNA, and these are small interfering RNA. So it is going to bind to the messenger RNA or to the DNA itself to block it. It helps to regulate gene expression or to cut it up. The last type is called a ribozyme. And a ribozyme is a type of RNA, and it has a job just like an enzyme. How is the genetic code read? So for each gene, one DNA strand is read, and this strand is known as the template strand. The RNA that is going to be synthesized or transcribed from that DNA, again, it's called mRNA. And mRNA is assembled from the 5' to the 3' end, and it's going to be complementary to the original DNA template. So if you look at the picture on the right, you can see the DNA template strand. What is being read is the 3' to 5' turquoise color DNA half at the top with the code ACCAAA, etc. The complement to the A is a U. Typically, A's go with T's, but because we are building RNA, you can't have a T nitrogenous base. You have to have a U. So the complement to an A is a U. The second letter in that top strand is a C. The complement to a C is a G. And the third letter is a C, so the complement is a G. The 3 letters together, the RNA triplet, is also known as a codon. It is this 3-letter code or codon that you are going to use a table to figure out what amino acid is going to be in that polypeptide chain. So I don't have the chart available on this slide, but if you look at the first codon in that RNA strand, it is UGG. If you look at the chart and when you find the 3 code UGG, it is going to code for tryptophan or TRP, which will be the first amino acid in this example of a polypeptide or protein. So here's the chart in which you will use to look at your mRNA codon, and you will be able to figure out what amino acid is going to be in that polypeptide or protein. So there are 64 different codon combinations of these letters, of these nitrogenous bases. Again, A's, U's, C's, and G's. Now, there's a little bit of repeat or redundancy. Sometimes you're going to have more than one 3-letter codon for an amino acid. And please note that you must read the mRNA strand in a reading frame of 3. So very quickly, I want to do an example. So this would be an example of a piece of DNA. Again, we only look at the template strand, one side of the DNA molecule. We're only going to look at the 3' to the 5', because mRNA is built from 5' to 3'. So when I want to figure out the mRNA, the complement to these letters, the first letter that I look at, or the first codon, is TAC. The complement to TAC is AUG. The second codon in this strand is CGC, and the complement would be GCG. And finally, the last codon is AAA, and we don't use T's as a reminder in the mRNA strand, so the complement would not be TTT, but it would be UUU. Then you are going to look at the chart, and when you look at the chart, you want to start on the